Can somebody confirm that you can hear me? The Zoom didn't start right. Like it said the video was off and the speaker gave me a warning.
I can hear you.
Thanks.
I can hear you.
All right, before we begin, are there any questions? Oh, I need to put my thumb drive in.
Hi, I have a question. Are you going to make the PDF for chapter 2 available?
Have we finished chapter 2?
We have. Yeah, we started
Yeah,
chapter
then I'll
3
make it
last Thursday.
available.
Awesome. Thank you.
Uh, Let me see. Let me remind you, you need to clear a project on the infectious disease project with me. So pick a topic and then request it with me, and I'll tell you if it's possible. You have to be the first person to request that topic. And I also want to make sure that it's actually an infectious disease that you can write a report on.
I have a question.
Share my screen. Yes,
The quiz tomorrow, is it chapter 1, 2, 3, and 4, or just the first 3 chapters?
I have that right here. Um, Today is July 23rd. The quiz will be July 24th. It's going to cover chapters 1 and 10, the start of chapter 2, and then labs 0, lab 1, and lab 2. Okay?
Thank you.
All right. So we'll talk about the lab later today. And it looks like we're supposed to be on chapter 3. I don't know if we started chapter 3 or not. Let me open up chapter 3 and then I'll look at make chapter 2 available to you.
Isn't chapter 3 regarding the microscope?
Yes.
Yeah, we started that.
Yeah, we started it. I know we did in the lab, but let me see. There's chapter 2. All right. Oh, yeah, I remember pointing out the metric practice quizzes. So let's go to chapter 3. Said we had finished slide 5, showing you the different things you can see with different, like the human eye, the light microscope, and the electron microscope. I thought I talked a little bit about this, but maybe that was in the lab. Let me blow this up. It's cooler today. You might notice I'm wearing a sweatshirt. I was actually a little cold this morning. Uh, so with the light microscope, and we're talking about a compound microscope, that is a microscope that has 2 magnification lenses, the objective lens right here and the ocular lens right there. The total magnification is the magnification of the objective lens times the magnification of the ocular lens. Typical microscopes and all the microscopes we have in the microbiology lab at Clark College has a 4x objective lens, shown right here, a 10x objective lens, sort of shown there, 40x objective lens shown there, and then 100x or oil immersion lens shown there. You should know these parts of the microscopes. The ocular lens, also called the eyepiece, The objective lens is down here. It's on a rotating nose piece, which isn't shown, but it's right there. So that you can rotate these lenses around. The body tube, which you don't really need to know, but you probably should know the arm. You can carry the microscope by holding on to the arm and one hand under the base. The stage where you put the slide. Stage clip, which isn't marked here, but you should know it, is what holds the stage in place. The condenser lens, and then the iris diaphragm. And I've never figured out why on some microscopes the iris diaphragm is above the condenser lens, and then in other microscopes, the iris diaphragm is below the condenser lens. The iris diaphragm can open and close, giving you a cone of light to where the slide is. You want the cone of light to be about the size of your field of view. And so the iris diaphragm should be fairly open, meaning open most of the way with the 4X lens, and then shut down most of the way with the 100X lens. Any question about that? The condenser is a glass lens, but all it does is it gives a cone of light, focusing it on where the slide position would be, where the specimen would be. It does not magnify the specimen. The illuminator is just the lamp. Coarse focusing knob, the big knob, the fine focusing knob, the little knob, and then the base of the microscope here. And you should know the light switch. On this one, there's only one switch. And to turn it off, you just rotate that knob all the way down, and then it'll click and it'll click off. And then you turn it on, you move it past the click. And then to turn the lamp on higher, you turn it further. So on this one, the rheostat and the on-off switch is in the same switch. Any question about any of this? This is a simple microscope. It's actually a microscope built by Antony van Leeuwenhoek. And it's a simple microscope because it only has one magnification lens. So in reality, a simple microscope is nothing more than a super duper magnifying lens. Because if you were to have just one magnifying lens, it would be very similar to this. The difference is that this is a microscope, although this is a primitive, one of the earliest microscopes ever built. You don't need to know the parts to this. Okay, when we're talking about microscopes, it's important to understand the concept of resolution. The resolution is the ability of the lens to distinguish two points as separate points. And if you look at them and the 2 points are close enough together, they will actually look like one point. And I'll show you that in just a minute. Here you can see 2 points. This is the actual, I don't know, artist's rendition. And this is what it would look like under the microscope. And these points here, you cannot distinguish them as separate points, or those two there. And so these points are within the distance for resolution, meaning they have to be separated by more than 200 nanometers to see two distinct points. Any question about any of that? So the light microscope with the resolving power of 0.2 micrometers, that's 200 nanometers, can distinguish between 2 points that are greater than 200 nanometers or 0.02 micrometers apart from each other. If the 2 points are closer, you cannot distinguish them as two separate points. The best compound light microscopes can magnify a total of 2,000 times. That'll be a objective lens of 100x and a ocular eyepiece of 20x. and the best that you can get will be 2,000 times that can distinguish 2 points at 200 nanometers apart from each other. If you were to increase the magnification, it would be blank magnification because the resolution limit would not change. It would still be 200 nanometers. And that's because that size is actually dependent on the size of the wavelength of white light. Is that clear to everyone? So even if you were to build a higher magnification microscope, it will not magnify any further. It would just be blank magnification because the resolution limit is not dependent on the microscope. It's dependent on the wavelength of light. Any question about that? Now, remember that our microscopes are not the best microscopes. They only have an eyepiece of 10x, and you could magnify a little bit further on our microscopes. Trying to just say what we do here. Like I said, if you were to magnify any further, you'd still see this. And that's because it's not dependent on the microscope. It's dependent on the wavelength of light, white light. Light also refracts, and let's talk about refraction with a microscope. Refraction is the light bending ability of a medium, and different media has a refractive index. For example, it looks like this pencil is bent, and that's because light leaving the water will refract or bend when it gets into the air. So air has a different refractive index than water, making the pencil look bent, and in artist's rendition, it sort of looks like that. Refraction is very important for life because when a bird sees a fish in this position, the fish is really in this position. And birds instinctively know that if it sees the fish here, it has to put its feet down here to catch the fish. Any question about any of that? So light has a refractive index that's different in air and light. And when we are going through the light source here, actually the light's going to bend right here to the glass, but we are not showing that. But we're showing the light ray leaving the glass. And you can see that some of the light rays bend significantly, meaning that light ray is not even going to hit the objective lens. Any question about that? And this is showing you some different light rays coming through air, passing through glass. Some of the light rays will not be refracted, and they will be straight, going from the lens through the air and to the glass of the objective lens. This one here will be bent slightly when it leaves the glass coming into the air, and then it'll be bent once again when it changes from the air to the glass of the objective lens. The objective lens can gather this light ray and that light ray, but it cannot gather this light ray, which is bent further, and it's not going to be caught by the objective lens. And of course, it doesn't gather this light ray either, and that light ray is hit so hard or bent so hard, it doesn't leave the glass. It's bent back into the glass. Any question about that? And just different light rays have different, I don't know, bending angles, we'll word it that way. If you use oil, which has the same refractive index as glass, then none of the light rays will be bent. So that one won't be bent. Come on, mouse. That one won't be bent. That one won't be bent. And you can notice that when you use oil, you gather more of the light rays, meaning you gather that one, and with no oil, you don't gather that one. So if you use oil, you improve the resolution of the microscope because you're gathering more light rays. Any question about any of that? And that's why immersion oil is used with the 100x oil immersion lens, is to stop the refractive bending of light when light leaves the slide, the glass slide. And that will improve the resolution and magnification of the specimen. Any question about any of that? All right. Bright field illumination is what we normally have with a light microscope, where the light comes from a lamp source, goes through the condenser lens focused on the specimen, and then is gathered by the objective lens. Both the specimen will be light, and then the background, meaning the glass of the slide, will be very light, because all you're seeing there is the light coming from the lamp. Any questions about bright field illumination? You need to understand how it differs from dark field illumination. Dark field illumination, we have an opaque disc in between the specimen and the light source, the lamp. And so light directly coming from the lamp going through the specimen is blocked. The only light that we get from the lamp to the specimen to the objective lens is light which comes through from the lamp, obviously, but it's at an angle such that the only reason why it's collected by the objective lens is because this light is being refracted from the specimen. So the light is being refracted from the specimen, and some of that is refracted at the correct angle to be collected by the objective lens. Is that clear to everyone? We don't have light directly coming from the lamp, going through the specimen, going to the objective lens. We only have light rays at a strong angle coming to the specimen, and the refraction of the light bends it into the objective lens, causing dark field illumination where the specimen is dark, but it's light compared to the background. The glass of the slide is totally dark because it gets no light from the lamp. It's blocked by the opaque disc. Any question about any of that? All right, you should know the difference between bright field illumination and dark field illumination. You don't have to know the intimate details, but you should know how they differ. That's enough for light microscopes. Let's talk a little bit about the electron microscope shown here. The first thing you can notice is an electron microscope has a couple of differences with the light microscope. And one difference is the electron microscope is a lot bigger. Another difference, which is a little more important, is the electron microscope does not use light rays. It is using beams of electrons to magnify the specimen. So it doesn't use a glass lens, it's using an electromagnetic lens which focuses the beam of electrons onto the objective lens. The shorter wavelengths of electrons gives greater resolution. You don't need to know this, but the wavelength of an electron is 100,000 times smaller than the wavelength of visible light. This allows examination of very small specimens, such as a virus and the internal cell structures. There are two main types of electron microscopes. There's actually 4 types of electron microscopes. You need to know the two main types. The transmission electron microscope shown here and the scanning electron microscope shown here. What do you notice between the two differences of what you're seeing in the different types of electron microscopes?
You could see the organelles on the one on the left.
Yeah, you can see the internal structure of the cell on the transmission electron microscope, which in this case are organelles. And on the right, what are you seeing? You're not seeing inside the cell.
Is it the outside of the cell you're seeing?
Yeah, you're seeing the surface of the cell, the outside of the cell. And you'll notice that this is also the scanning electron microscope is showing you the image in 3 dimensions. The transmission electron microscope, you're only seeing the image in 2 dimensions. Any question about any of that? So with transmission electron microscope, you're only seeing a plane through the cell. With the scanning electron microscope, you're seeing the cell in 3 dimensions, or the object specimen in 3 dimensions. This is another scanning electron microscope, and you can see the 3-dimensional structure of this crystal. And here is a transmission electron microscope. I believe that is the membrane of a chloroplast. And you can see the structure of the chloroplast membrane. Membranes, I guess we should say, right there. 2 other types of electron microscopes which you do not need to know for the quizzes is a scanning tunneling microscope. Using a metal probe to scan a specimen, you can get a resolution of 1/100th of an atom with a scanning tunneling microscope. And let me move that out of the way here. This is actually a protein from E. coli. And then the 4th type is atomic force microscopy. You also will not be quizzed on this type of electron microscope. Using a metal and diamond probe inserted into the specimen, it produces a 3-dimensional image of your specimen. Here we're looking at the toxin for Clostridium perfringens. And I guess I should mention that with the scanning tunneling microscope, we can magnify the greatest of all the electron microscopes. 1/100th of an atom. The hydrogen atom is pretty small. All right, here we're looking at 2 slides looking at cells here and here. Let me blow that up a little bit. But my question for you is, which cells are easier to see? The cells on the left or the cells on the right?
The ones on the right.
The ones on the right. The only difference between this slide and that slide is these cells are stained. Staining, by adding a dye to the cells, all it does is it colors the cells. In this case, it's a dark color. And all it does is increase the contrast between the cell and the glass background of the slide. And here you can see the cells are essentially transparent and they don't have much contrast with the glass and the liquid of the slide. Any question about that? So we normally add a stain to the slide to make the cells show up better. And the stain, all it is doing is increasing the contrast between the cell and the background of the slide, meaning in this case, the glass of the slide. So most microbes are colorless and have little contrast with the slide or medium. To see them, you really need to stain the cells. So staining is coloring the specimen or the background to increase the visibility and the contrast between the cells and the background of the slide. Now, before you do the staining, you need to make a smear. And we usually call that a bacterial smear because in this class, almost all the smears you make will be a bacterial smear. A smear is a thin film of cells, microbes, on a slide. And so before you do the staining, you make the smear. Is that clear to everyone? The smear is something different from the stain. Usually, the smear will be dried, air dried, and then heat fixed to attach the cells to the glass of the slide. Without air drying and heat fixing the cells to the glass of the slide, they will come off just by adding the liquid of the stain. And then they'll wash off when you rinse the slide. Any question about that? So usually you air dry the specimen and then heat fix it by putting the slide gently through the Bunsen burner. You just take the slide like this, hold it with your hands, and then here's the flame. Just pass it over the flame 3 times. And you do not want the slide to get so warm that you can't hold it, meaning it's not supposed to burn you. I usually tell my students always use your hands because if you use a slide holder, you won't know that you're getting the slide too hot. And if you use the Bacto incinerator, you usually just pretend this is the Bacto incinerator. You just put the slide either right above where the heat source is coming out, or you can actually touch the slide on the heat source briefly, just for a second or less. And you do that 3 times. Stains or dyes, both words are used, are salts consisting of a positive and negative ion. In a dye, one of the ions is colored. We call that the chromophore. A basic dye is where the chromophore, the coloring part of the dye, is in the cation, or the positive charge of the salt. In an acidic dye, the chromophore is in the anion, or the negative part of the salt of the dye. Any question about any of that? Most of the dyes in microbiology are basic dyes, but there are a number of acidic dyes. Bacteria have a slightly negative charge, therefore basic dyes are better for staining the cell. Some basic dyes include crystal violet, methylene blue, malachite green, and safranin. Some acidic dyes, they typically stain the background of the cell and not the microbe, and that's because the acidic dye has a negative charge, and if the bacteria cell has a negative charge, the dye will be repulsed from the cell, and that will stain the background of the cell, not the slide. Eosine, acid fuchsin, and nigrosin are 3 acidic dyes. We call an acidic dye that stains the background of the cell negative staining. If you remember or know anything about Polaroid films, there was a negative to it, which showed an object in black and white, and it was negative to the coloration of the specimen. So if you saw the face, like the whites of the eye would be black, and then the pupil would be white in the negative stain, and the negative. Well, that's the way the negative stain is as well. showing the cell in, I guess, reverse contrast. Any question about any of that? Here is a cell stained with a basic stain, and there are cells stained with an acidic stain or negative stain. And both of them, you can see, show the cells quite well, even though this is staining the cell and this is staining the background. Any question about any of that? All right. There are three main types of stains we have in microbiology: a simple stain, a differential stain, and a special stain. A simple stain just uses one dye, meaning a single dye, and it's either in an aqueous or an alcohol solution. The primary purpose is just to increase the contrast between the cell and the background of the slide, or the background of the cell. The stain is applied to a fixed bacterial smear for a certain length of time. It's usually then dumped off. It may be washed off, dried, and then examined under the microscope. Any questions about simple stains? Whenever you use one stain, it is a simple stain, and it will stain all the cells the same color. Any questions? Sometimes a mordant can be used with the stain. It intensifies the staining, like when we use iodine in the Gram stain. Gram's iodine functions as a mordant. It is not a stain. It increases the affinity between the stain and the specimen, meaning Gram's iodine actually binds to the crystal violet and then stains the cell more intensely than the crystal violet alone. You can use a mordant also to coat the specimen to enhance it. Like in the flagella stain, we use a mordant which puts down many layers of the mordant on the flagella, making the flagella easier to see. So those are two examples of a mordant. You really only need the one where we're talking about Gram's iodine being a mordant in the Gram stain. It is not a primary stain or even a secondary stain. It is a mordant. A differential stain is a stain that reacts differently with different kinds of microbes. Therefore, you can distinguish between two different types of microbes. Usually, a differential stain uses two dyes, but this is not required. Most commonly used differential stains in microbiology are the Gram stain and the acid-fast stain. Both of these stains use two dyes in the stain. So the Gram stain is a differential stain which was developed in 1884 by Hans Christian Gram. You don't need to know the year that he invented it, but you should know that the Gram stain was invented by Dr. Gram, and that's why when you write it, you should always make that a capital G, because it's named after a person. Any question about any of that? Dr. Gram actually did intend it to be a differential stain, but he intended it to differentiate between the bacterial infective cells and the cells of the patient, meaning when he used it, the bacteria were staining dark purple. We call that Gram positive today. And then the cells of the patient were staining pink, or Gram negative today. And then microbiologists started using Gram stain, and they discovered that it was a very useful stain in bacteriology because it could differentiate between almost all bacteria into two different types of cells, the gram-positive cells and the gram-negative cells. The gram-positive cells stain dark purple. The gram-negative cells stain pinkish. Any question about that? The difference between these two cells is based upon the differences in their cell wall. How you make the gram stain, this is just a general one slide overview, is first, before you do the gram stain, you have to have a bacterial smear on a glass slide. And of course, you must allow the bacterial smear to air dry, and you must heat fix it. You absolutely must heat fix it to attach the cells to the slide. Otherwise, the gram staining procedure will wash all the cells off the slide. Any question about that? So the gram stain, actually steps 1 and 2 are not really part of the gram stain. It's steps 3 through 11, which are part of the gram stain. Any question about that? The bacterial smear is step 1, and then 1.5, you air dry it, and then 2, heat fix it. You then, the first thing you do with the cells, once they're heat fixed, is you flood the slide with crystal violet for 60 seconds. You then dump the crystal violet off and wash gently with cold water. You then wash the water off, meaning dump the water off, and then you flood with iodine. Iodine is a mordant. You apply it for 60 seconds. A mordant will intensify the stain of the crystal violet, and that's because the Gram's iodine actually complexes with the crystal violet, making a larger molecule than the crystal violet or the iodine alone. You then dump the iodine off and wash gently with distilled water, or not water, just with cold water. You then flood the slide with the decolorizing agent, just briefly for about 5 seconds or less. And usually the colorizing agent we use in the microbiology lab is acetone and ethanol, but it could be acid and ethanol, or it could be just ethanol alone. Those are the 3 different decolorizing agents. Like I said, the one we mostly use is acetone and ethanol. You then wash immediately with cold water. It has to be immediate here, because if you were to dump the decolorizing agent off, traces of the decolorizing agent will be on the cells, and that could pull out the color from the gram-positive cells, which you do not want. You only want the color to come out of the gram-negative cells, which you do want. Any question about any of that? So you wash immediately with cold water to remove the decolorizing agent. You then flood the slide with safranin. Safranin is a counter stain because it differs from the primary stain, and you apply that for 1 to 2 minutes. I usually use 1 minute because our safranin has always been strong in Clark College. So 1 minute's good enough. And then you can remember you're just applying everything except for the decolorizing agent for 1 minute. And then after 1 minute or 2 minutes, you wash the slide with cold water, then blot it dry. Let me explain a little bit about that. You get bibulus paper, which is really thick paper, almost thick like cardboard, and then rip it out and then bend it or fold it over the slide. My phone is the slide here. I don't know if you can see this. And then just blot it dry. Do not wipe the bibulus paper over the slide like this. Do not wipe the slide. Just blot it dry. And that will remove the worst of the water. That will remove enough of the water so that you can then put even oil on the slide and look at the slide under the microscope. meaning the trace amounts of water will not interfere with your making an observation, even with oil, under the microscope. Any questions about the overview? If not, let's go into this in more detail. With the gram staining procedure, close that off, we're not there. You apply crystal violet, it's the primary stain. All the cells stain purple. You then apply Graham's iodine. It's not shown here, but the Graham's iodine actually makes the cells a little darker and also a little more purple. This is a blue purple color. This will be a more purple color. You then flood this cell or treat this slide with the decolorizing agent. If it's done correctly, the gram-negative cells will then become clear. The gram-positive cells will retain the crystal violet Graham's iodine, and they will remain purple. You then flood the cell with safranin, and the gram-negative cells will then stain pinkish, and the gram-positive cells will stain purple. So you can see at this step here, the differential step of the gram stain is the decolorizing step, where the two different types of cells appear differently in color. Any question about any of that? So in a little more detail, you apply the crystal violet for 60 seconds. You then wash with cold water, and let me come up to the very beginning here. Remind me I'm on slide 35. Let's go back to the gram stain. After each step of the gram stain, you do have a gentle wash with cold water. And when you're giving me the steps of the gram stain, you don't have to detail a cold water wash after every step. You can just state after every step, there's a cold water wash. You can put it at the very beginning of the gram stain or at the end of the gram stain, or you can just put it in a wash with cold water and put something like, and do this after every step of the gram stain. Okay? Crystal violet is the primary stain, where crystal violet sticks to the peptidoglycan in the bacterial cell wall. It's considered the primary stain since it colors all cells the same. It's also the first stain you use. Well, it's also called the primary stain. The color of all the cells will be a blue-purple color. Gram-positive and gram-negative will be blue-purple. You then do the wash and then apply iodine. Remember, iodine is a mordant. It complexes with the crystal violet. You apply it for 60 seconds. After applying the mordant, both the gram-positive cells and the gram-negative cells will be a purple color, slightly darker than the crystal violet and a little more purple color than the blue-purple of the crystal violet. You then apply the decolorizing agent. In this slide, we're using acetyl ethanol for the decolorizing agent. It washes the crystal violet Graham's iodine out of the gram-negative cells, but if it's done correctly, it does not wash it out of the gram-positive cell. So it is considered the differential step in the gram stain, because here at this step, the gram-negative and the gram-positive cells will stain differently. Now it says to apply for about 5 seconds, but the best way to apply the decolorizing agent is to put it on and then watch the blue stop streaking. Once the blue stops streaking, you stop applying the decolorizing agent. And that will remove the crystal violet iodine from the gram-negative cells and not from the gram-positive cells. If, on the other hand, you were to continue applying the decolorizing agent, you will wash the dye out of the gram-positive cell. And that's why the decolorizing step is the trickiest and hardest step to do in the gram stain. And like I said, you apply it only for about 5 seconds, but the best way is to apply it until the blue stops running. And then wash immediately to remove the decolorizing agent, even the trace of it, from the gram-positive cells. So the color of the gram-positive cells will be a purple color if the decolorizing step is done correctly, and the gram-negative cells will be colorless. You then flood the slide with safranin. Safranin is a counter stain. It's a counter stain because it's a different color than the primary stain. And then wash with distilled water later. And that will change the color of the gram-negative cells to pink, and the gram-positive cells will stain purple. Now let me mention that safranin will stain all of the bacteria. If it stains all of the bacteria, why do the gram-positive cells not pick up the pink color? Anyone have a guess there? If you were to look at the cell with the electron microscope, and let's make this square right here the cell, you will notice that there is purple in different regions of the cell, like right here, and let's call that blue purple. And there is also pink in regions of the cell, but you can't see the pink unless you use the electron microscope. Under the light microscope and with your eyes, the cell will only look purple, and it's simply because purple is a darker stain than the pink stain, and our human eye cannot see the pink. It's hidden by the purple color. Any question about any of that? So when you stain the gram-positive cells with safranin, they do pick up the safranin. It's just the human eye cannot see it because the purple is masking the pink. Like I said, if you were to blow up the cell in an electron microscope, you can actually see regions of the cell which would be staining pink and other regions of the cell which will be staining purple. You then blot the side with bibulous paper, which will remove enough of the water that you could look at the cells under the microscope, even if you put on oil and use the 100x objective. All right, the gram-positive cells are purple. The gram-negative cells are pink after the gram stain. Why? What's the basis of the differential staining? It has to do with differences in the cell wall structure of the bacteria cells. The gram-positive bacteria cells have a thick peptidoglycan layer. It is thick enough that the grams iodine crystal violet complex does not wash out of the peptidoglycan with the decolorizing step. And the gram-negative bacteria do have peptidoglycan, or most of them, but it is much thinner. And the grams iodine crystal violet complex can wash out of that thinner peptidoglycan layer. Any question about any of that? So there's the gram-positive cell. Let me blow this up. Showing you the thick purple here, which is the peptidoglycan in the cell wall. The yellow is the cell membrane, lipid bilayer. Peptidoglycan layer is much thicker than the peptidoglycan layer of the gram-negative cell. I'll get to it in a minute. And it's showing you only 3 layers of peptidoglycan. In reality, it's more than 20 layers of peptidoglycan, and it may be over 100 layers of peptidoglycan. Meaning the artist has only drawn in 3, but in reality, it's 20 layers or more, up to over 100 layers. And the gram-negative cell is a much thinner layer of peptidoglycan. I don't even see purple in there, but there it is there. It's surrounded by yellow, which is a lipid bilayer, the cell membrane below it, and a lipid bilayer above the peptidoglycan layer, which we call the outer membrane of the cell wall. Both of them are lipids. They're both membranes. What else do I want to say? But this peptidoglycan layer is much thinner than there, and the iodine crystal violet complex washes out of this thinner peptidoglycan layer. The artist has only shown 1 layer of peptidoglycan. In reality, there's always at least 3 layers of peptidoglycan, up to about 20 layers. Any question about any of that? So that's the main difference between the gram-negative cells and the gram-positive, the thickness of the peptidoglycan layer. Oop, back. So crystal violet iodine complex forms in all cells, both the gram-positive and the gram-negative. The complex is larger than the crystal violet or the iodine alone. The crystal violet iodine complex is washed out of the thinner gram-negative cells because it's a thin peptidoglycan layer, and the lipopolysaccharide layer of the cell wall is destroyed by the alcohol wash. The gram-negative cells then appear colorless. The gram-positive cells retain the crystal violet iodine complex because the thicker peptidoglycan layer retains the crystal violet iodine, at least for a longer period of time, and so it does not wash out when the gram stain is done correctly, where the decolorizing agent is applied for 5 seconds or less. And the cells therefore stain purple. So here's a gram stain. We can see gram-positive rod, gram-positive conchi, more gram-positive conchi, gram-negative bacilli, gram-negative spirals. I don't see any gram-negative conchi, but there might not be any in this slide. Any question about that? Oh, there's a gram-negative vibrio. Come on, there we go. When you do the gram stain, you'll see things like this. And the gram stain is actually a hairy procedure, meaning you might be pulling your hair out because it is fairly tricky and difficult to do. And then there are problems like this, that, and that. How do you interpret this slide? Those are kind of a light purple, and those are very intense purple. These are bad regions of the slide. Let's see if I got another one there. Nope. This is actually a crystal of crystal violet. Another crystal of crystal violet, another crystal of crystal violet. And you can sort of see, let me blow this up a little bit, that these cells right there are staining darker than these cells over there. And that's because the crystal violet comes out of the crystal and then start staining the nearby cells. And what you want to do for reading this slide is, oops, didn't mean to do that. There we go. Is you want to read the good regions of the cell where there are not clumps and there are no crystal violet crystals. So don't read the cells in these regions, bad regions of the slide. Read it in the good regions of the slide. And sometimes you'll note that these cells and these cells and these cells will stain pink, but then these cells and these cells will be staining purple. And once again, like I said, the crystal violet just comes out of the crystal and then stains more intensely the nearby cells from the crystal. So don't read it where the crystal of the crystal violet is. And you sometimes have crystal violet crystals on your slide just because the crystal violet crystals form in the bottle of crystal violet, and usually they precipitate to the bottom. But if you happen to take the stain down at the bottom of the slide, or you have it in an eyedropper and you turn it upside down, the crystals will come to the top of the eyedropper, and then you put those on your slide. Gram stain, you'll learn, you'll start pulling out your hair because it doesn't work perfectly. You get the most consistent results when the gram stain is performed on young growing bacteria. In fact, let me state that when you're applying the gram stain, it should only be applied on cells that are 2 days old or less. If the cells are older than 2 days old, you may not get correct results from the gram stain. Some bacterial cells stain poorly or not at all with the gram stain, and that's usually the mycobacteria. They have a lot of mycolic acid in their cell wall. Mycolic acid keeps water from leaving the cell or entering the cell, and the gram stain, at least the crystal violet and the iodine, is soluble in water. And so these cells do not stain well. You can say they stain poorly. I've never had mycobacteria that did not stain at all. It always stained lightly. But there might be some that do not stain at all. And then the results are not always clear-cut as the theory suggests. That will become evident if you were to actually do the unknown project and then stain your cells with the gram stain. Fortunately for you, you will not be doing it because everything we do will be done online. I'll just send you a slide, a picture of the slide, which will be the your cells stained with the gram stain. And obviously I won't give you a slide where you have to interpret the slide, meaning there will be one region which will be pink and one region will be purple. I'll send only a picture with one stain or the other, meaning gram positive, gram negative. Any question about that? So most of the time when students get inconsistent results on the gram stain, I ask them how old their cells were, and the cells were always older than 2 days old. When you're using the gram stain, your cells can only be 2 days old or younger. If they're older than that, you should simply regrow your cells and then apply the gram stain. The reason is that the cells which are older than 2 days old will have some cells which are decaying or dead, and they may not stain correctly with the gram stain. They will oftentimes stain gram negative. And then if the cells which are viable meaning still alive, and they will stain correctly, and they will stain, or they may stain gram positively. All right, the structure of the gram positive and gram negative cell walls not only affects the gram stain, it also is related to how the cells respond to certain antibiotics. Gram positive bacteria tend to be killed easily by penicillin G, cephalosporin, and many detergents. The reason being is penicillin G, cephalosporin, and many detergents can get right through the cell wall and then hinder the cell, meaning kill the cells. The gram-negative bacteria are more resistant to these antimicrobials, such as penicillin G, cephalosporin, and some detergents. The reason is the gram-negative cell has an outer membrane of the cell wall. It does not allow penicillin G to enter the cell. Penicillin G cannot get into the cell, cannot get into the cell wall, it can't damage the cell wall, and then penicillin G will have no effect on the growth of those cells. And cephalosporin is similar. That doesn't mean that all gram-negative bacteria are resistant to all antibiotics. We have different antibiotics which we can treat gram-negative bacteria. For example, ampicillin can work on gram-negative cells as well as gram-positive. But penicillin G cannot work on gram-negative cells. And then we have streptomycin, an antibiotic which is lipid soluble. It will get through the cell wall of a gram-negative cell. So it may kill gram-negative cells better than it kills gram-positive cells. So streptomycin is an antibiotic, works very well on gram-negative cells. Any question about any of that? All right, you're not going to be examined on special stains. I want to talk about it because you may need to do a special stain for your unknown project, meaning I'm going to give you an unknown bacteria and you're going to try and figure out what it is. You may want to do the capsule stain on those unknown cells. The capsule stain is used to stain for the bacteria capsule. When you use the capsule stain, it stains the background of the cell, does not stain the capsule, and it may stain the cell. This capsule stain stains the bacteria cell very well, very darkly. And then it stains the background of the cell lightly, but the capsule is not being stained. With this capsule stain, it's actually India ink, it stains the cells light gray. And then the capsule is not stained, so it's white or clear. And then the background of the cell or the slide is very dark, darker than the cell. Any question about that? And that's India ink. All right, if there's no questions, that's it for Chapter 3. Let me see where we're supposed to go today. We are supposed to start Chapter 4. I suppose I should save down Chapter 3. And then close it down. All right, Chapter 4, the functional anatomy of prokaryote and eukaryotic cells. We're going to be discussing the functional anatomy of prokaryotic cells. We will also talk about the endosymbiotic theory for eukaryotic cells. However, I'm only going to give you the slideshow of the anatomy of eukaryotic cells. You should have had that in your Biology 160 class or the precursor class to microbiology. So this should be a review for you. And I never have the time to talk about it, the functional anatomy of eukaryotic cells, especially in the summer, because the summer is more truncated than any other term. So I know I won't get to the functional anatomy of eukaryotic cells. Any question about any of that? So in this slide, we have the major goals and the rough outline. Know the terms. What's a coccus, a bacillus, a sporilla? What's a streptococcus, staphylococcus, diplococcus? What's a tetrad? What's a Sarxinia? What's a vibrio? What's a spirochete? What's the glycocalyx? What's a flagella? What's an axial filament? What's a fimbriae? What is a pili? What is peptidoglycan? What is endotoxin? What is selective permeability, diffusion, facilitated diffusion, osmosis, active transport, tonicity, plasmid? What's a plasmid? What are inclusions? What's an endospore? And what's the endosymbiotic theory? Now, these are only the major goals. To get all of the goals, you need to look at the objectives to chapter 4. Well, that's odd. I've got this one separated into objectives and chapter notes. Any question about any of that? All right. We'll also talk about how gram-positive and gram-negative cell walls differ, and you need to be able to describe the importance of taxis in bacteria. When we're looking at prokaryotes, comparing them to eukaryotes, prokaryotes have they lack a nucleus, and prokaryote is actually Greek for before pre-nucleus. Literally, that's before kernel, and they use kernel for the nucleus. And then the Greek word for eukaryotes is true nucleus, literally true kernel for true nucleus. The prokaryotes differ from the eukaryotes, the bacteria. There's one circular chromosome usually, and it's not in a nucleus. For eukaryotes, they have linear chromosomes. There's multiple chromosomes, and they're in the nucleus. They also differ in the presence of histones. The DNA of eukaryotes is associated or wrapped around a protein that we call histones, and that protein is missing or absent in the prokaryotes. The bacteria and the archaea lack membrane-bound organelles, which I usually just call organelles. The eukaryotes have membrane-bound organelles. A membrane-bound organelle is an organelle which is surrounded by a membrane, such as the Golgi complex, the mitochondria, and the chloroplast. You could call an organelle a non-membrane bound organelle. I just don't use that term for any organelle that does not have a membrane around it. And that would be something like the, what is that called? Sorry, I'm drawing a blank here now. Well, a ribosome would be an organelle without a membrane around it. A mitotic spindle, what is that? It's made by the, forgot the name of that organelle. Sorry, drawing a blank there. I'll remember it. Just give me a little bit of time. Anyways, a ribosome is an organelle. Centriole. A centriole is a non-membrane bound organelle found in animal cells. There are no membrane bound organelles in a bacteria, but there may be non-membrane bound organelles like ribosomes are present in a bacteria. Peptidoglycan is found in the cell walls of bacteria only, so this molecule is only found in bacteria. No peptidoglycan is found in the cell walls or any other part of the eukaryote cell. Prokaryotes, most of the cells divide by binary fission. Eukaryotes, most eukaryote cells divide by mitosis. Another way that eukaryotes differ from prokaryotes. We're going to talk about the cell shape and the cell arrangement of prokaryotes. We'll talk about the cell structures and then the movement of material across membranes. Eukaryotes, we won't really get into it, except for when we're talking about the endosymbiotic theory. So prokaryotes have a cell size and a cell shape. The average bacteria is about 1 to 2 micrometers in length or in diameter, doesn't matter in this case. The smallest bacteria is about 0.4 micrometers long, up to about 10 micrometers long. You can get bacteria smaller than 0.4 micrometers, but it's thought that those cells which are smaller than 0.04 micrometers down to about 0.2 micrometers, it's thought that those cells are non-viable. We have 3 different basic shapes for prokaryote cells. There's the coccus cells, and they're round. There are the bacillus cells, and that's shaped like a rod. And then there's the spiral cells, shaped like a spiral. We have 3 types of spirals, and the first type, the vibrio, is a half a spiral. You could also call it a bent rod. So either name is correct, a bent rod or a vibrio or half a spiral. And then there's the spirillum, which is a spiral. It's shaped like an S. Is that an S? Sort of. And then there's a spirochete, which has many more bends to it. The spirochete tends to be less rigid and more flexible than the spirals. Any question about that? The prokaryotes, we can have some cells with unusual shapes, like the star-shaped stela. And I don't know why they call it a square haloarchaea cell, but actually it's a rectangle. It's clearly a rectangle, not a square. And let me go on one more. There, they have it here, and it shouldn't be in this slide because we're talking about the cell arrangements in this slide. Coccobacillus is not a cell arrangement. It is a cell shape. And that is, if you have a rod and you make it smaller and smaller and smaller, let me try and blow that up. It'll eventually become so small that you no longer have a rod shape, you have a coccobacillus. And that's a cell in between a rod and a coccus. That one's clearly a coccobacillus. That's a really, really short rod, that one there. Any question about that? That cell's about to divide, that one there. So the Coccobacillus shouldn't be in this slide because it's not a cell arrangement. It is a cell shape. And it's sort of a slightly unusual shape for a bacteria, but it is also a part of the three shape, meaning there's three main shapes, coccus and bacilli, and this is both a coccus and a bacilli, the Coccobacillus. Most bacteria species are monomorphic, meaning they have one cell shape. Like the Stella is all star shape. The Staphylococcus aureus or Staphylococcus epidermidis is all cocci. But there are some species which are not monomorphic, which means a single shape. There are a few species which are pleomorphic, where the cells have many different shapes, shown here. Oftentimes, if the species is pleomorphic, the bacteria will lack a cell wall. And that's why they are pleomorphic, and that is whatever the cell shape is, it's just what the cell membrane is for those different shapes. And that's often seen in cells that do not have a cell wall. They'll be pleomorphic. But there are a few exceptions. All right, let's talk about cellular growth arrangements. These are how the cells stick together when they are growing. The first cell arrangement is actually shown right here, and that's a singlet. It's not listed here, but a single cell. We can actually see a singlet right there, as well as there, where you have a single cocci, and when it divided, no cells stuck together. So it's a singlet. And then you can have diplococci, which would be two shown right there or right there. And you'll note the growth plane of division is always this way. And if this were to divide here and here, you then get four stuck together. And that would be part of a streptococci, where the cells grow in a string, one hooked to the other, streptococci. And there's a streptococci right there. You can also have bacteria growing as a foursome. We call that a tetrad, or bacteria cells grown in a sarxinia right there, and it's really hard to see that. Kind of right there, and right there. Our sarxinia, of course, you can't see all of them because they're hidden behind another cell. Like this one right there, you can only see 7 of them, but there would be one cell right there hidden by these 2, or maybe those 3. Any question about that? You can also have staphylococci, and that's where the cells grow in a clump. Right here. Come on. There we go. Or right there. Staphylococci literally means cocci growing in a cluster looking like grapes. You put a stem on there, and that looks like a grape cluster. Any question about any of that? The cellular arrangement or cellular growth arrangement for cocci is more than for bacilli or for spirals. Bacilli only divide across one axis, so there are fewer arrangements. You can, of course, with a bacilli, have a singlet as a single bacilli, and that would be like this one here or that one there. You can have diplobacilli, and that's where the cell splits down here, and then grows into 2. Or streptobacilli, and that's where the bacilli is in a string connected on the short side of the cells. And this sort of looks like a sausage link. If anyone knows what I'm talking about. Apparently they used to put sausages and that type of a link like that. And Streptobacilli sort of looks like a sausage link. Coccobacillus is not a cellular growth arrangement. It is a cell shape, so it shouldn't be in this slide. And then the last cell arrangement of bacilli is a palisade. That is Streptobacilli, but they're not connected on their short end. They're connected on their long end. And palisade comes from the name of a fence. See if I can get, I'm not getting paint here. There we go. So the cells here grow like this. So this would be the dirt of the fence. And then you take the wood plank and you put them up like that. Anyone ever read Mark Twain? I believe it was Tom Sawyer was painting a palisade fence, and he convinced Huckleberry Finn that it was actually fun to paint the fence, and so he got Huckleberry Finn to paint the palisade fence instead of having to do it himself. Anyways, that's just a terminology. Palisade fences aren't very common anymore, but in the past they were very common. You should also note that bacillus has two meanings. There is the shape bacillus, which is a single rod, and we use the lowercase b to describe that. There was also the family Bacillus, and that is a family of bacteria, and it will have a capital B. Any question about that? And those two are the same. The difference is only between the capital B and the lowercase b. Do not confuse the family Streptococcus with the capital S, and then the Streptococci, which is a type of bacteria growth arrangement. And this is actually spelled a little differently. The Streptococci, the family is Streptococcus. All right, any question about any of that? If not, let's go on. When we're talking about the cellular growth arrangement, the cellular growth arrangement of bacteria will almost always be pleomorphic. So whenever you look at the cellular growth arrangement, you'll see more than one cellular growth arrangement. And for the bacilli, if it's a streptococci, the streptococci can break, forming diplococci. The diplococci can break, forming singlets. And so usually when you see streptococci, you'll see singlets, diplococci, and streptococci. And that's just because of the way the cells break. With staphylococci, you can usually see staphylococci, maybe Sarcsinia, maybe tetrads. Very rarely would you see streptococci and staphylococci, but it's possible. And then diplococci, and then singlets. With Sarcsinia or tetrads, you might see diplococci or singlets. And then with singlets, of course, you'd only see singlets. And that is the exception, and that is if the species only gives singlets, that's the only cellular growth arrangement you'll only see. And that is if the species only gets singlets, that'll be the only time the cellular growth arrangement is not pleomorphic. The Sarracenia, no. Sorry, I'm drawing a blank here. The spirochetes have a cellular growth arrangement of only singlets. So if you're looking at a species which forms spirochetes, you'll only see singlets, single spirochetes. Any question about any of that? All right. Some typical structures of a typical prokaryotic cell is shown here. First, you might have structures coming out of the cell. These are fimbriae and flagella. You'll note the flagella is much longer than the fimbriae. And another structure coming out of the cell would be the pilus or the sex pilus. It is also longer than fimbriae. Fimbriae sort of look like hairs. Flagella, the purpose of that is just to whip and then make the cell move. So if the cell can swim or move through liquid media, it has a flagella. Fimbriae, its main function is just to serve as an attachment to something. And then the sex pilus is actually, its main purpose is to allow plasmid DNA to move out of this cell and then move into another cell. So the sex pilus actually allows DNA to move out of one cell and move into another cell. Any question about any of that? All right, we have the cell wall shown here. The capsule, which is external to the cell wall, shown there, and that's a real thin capsule. And then below the cell wall will be the plasmid membrane. It's a lipid bilayer. We have cytoplasm inside the cell membrane, and that's for a prokaryote. Essentially, all of the cell is cytoplasm. The exception would be something that's big enough it does not dissolve in the cytoplasm, and that would be an inclusion. We'll talk a little bit more about that. Ribosomes. And really everything else would dissolve in the cytoplasm. There is no membrane-bound organelles in the prokaryote cell. You can have a nucleoid region. Those cells stain differently. I mean, the nucleoid region stains differently in the cytoplasm, but the only difference is the nucleoid region is wherever the DNA molecule is floating around in the cytoplasm. So there is no distinction between the cytoplasm and the nucleoid region because the DNA molecule can move wherever it wants to in the cell, and wherever the DNA molecule is, at least the chromosome, will be the nucleoid region, but there's no membrane between the two. And then bacteria may or may not have an extra chromosomal molecule of DNA that we call a plasmid. It's a small molecule. The chromosome is a very large molecule in the sense that it is a circular molecule. In that sense, it's the same, but it's a smaller molecule. We'll talk more about plasmids when we talk about chapter 8. Any question about any of that? Let me talk a little bit more about the inclusion. And the inclusion is something the cell is accumulating. And initially, when the cell is accumulating it, the cell won't have enough of it, so it will dissolve in the cytoplasm. But as the cell continues to accumulate whatever it's accumulating, there will become enough of it that it will actually precipitate out of solution from the cytoplasm. And then it will be this solid structure in the cytoplasm that we call an inclusion. Any question about any of that? All right, the structures found in red are found in all bacteria. There are 3 structures that are found in all cells: cytoplasm, plasma membrane, and then DNA. Those are found in all cells. So all cells have DNA, all cells have a plasma membrane, all cells have a cytoplasm. The glycocalyx is something secreted by a cell. So many prokaryotes can secrete this. It's outside the cell wall, so we call it outside the cell, meaning the cell wall is the last part of the cell that we call the cell. The glycocalyx is usually sticky. It can be one of 3 types of glycocalyx. And you could also pronounce that glycocalyx, glycocalyx. The 3 types of glycocalyxes is there can be a capsule. It's a neatly organized structure. It can prevent phagocytosis by a white blood cell. It does have water in it, but water is not the, how do I say it? It's not mostly water, like 95% or more water, the capsule. And the capsule can be seen by the light microscope. So if you're looking at a cell and you see a capsule under the light microscope, we call it a capsule because that's what a glycocalyx is if you can see it under the light microscope. If you can't see it under the light microscope and it's loosely organized, very loose, and mostly water, then we call it a slime layer. You cannot see a slime layer under the light microscope because it's mostly water. You need to have an electron microscope to see a slime layer. And then the third part of glycocalyx is the extracellular polysaccharide, which is made in biofilms. The bacteria inside the biofilm actually secrete material outside the cell, which creates the biofilm. And it's called the extracellular polysaccharide. Any question about any of that? Anyways, those are the 3 types of glycocalyxes. We'll mostly be talking about capsules because you can see it under the light microscope. And it's also, it aids the cell in being pathogenic because it prevents phagocytosis of the bacteria by white blood cells. All right, the flagella is the structure on a prokaryote that allows the cell to move. So it gives the cell locomotion. In eukaryotes, we have 3 structures which can give locomotion. We'll talk about those later. One of them can have flagella, but the only structure that allows the bacteria cell to move is the flagella. It is outside or mostly outside the cell wall. It is made up of the protein flagellin. And when we look at the flagella, most of it is outside the cell wall. There is a small portion inside the cell wall, right here, from here to here, called the basal body. of the flagella. You don't need to know that term. And there's even a portion of the flagella which is in the plasma membrane, this portion right there, which is, well, they don't have a name for that, but the last part of the basal body. Any question about the flagella? The flagella has 4 basic arrangements that the bacteria flagella can take. You don't need to know these names for the flagella arrangement, except for one of the labs will ask you, what is the flagella arrangement? So you need to know it only for that lab. I will not give you a quiz question on the bacteria flagella. But if the flagella are only on one side of the bacteria, we call it monotrichous. Let me blow that up so I can read it. Monotrichous, one flagella on one side of the cell. If the cell has 2 flagella on different ends of the cell, it's called amphitrichous, and there's only one on each cell, one on each side of the cell to be amphitrichous. If there's multiple on both sides of the cell, we then call it lophotrichous. And if there's a flagella coming out all over the cell, we call it peritrichous. And I'll mention that if you see cells with pleomorphic flagella arrangement, you always name it by the highest arrangement you see. And that's because a peritrichous flagella arrangement will be this, but the cell has to put out the flagella. So if the cell is still putting out flagella, it may not be peritrichous, it could be lophotrichous, or if there's only one on each side of the cell, it could be amphitrichous. And if there's only one flagella, it will be monotrichous. But then again, the peritrichous cell, after it first divides, it will not have any flagella, in which case there are no flagella. And that would be why the cell would look pleomorphic when looking under the microscope. It will eventually become peritrichous, but it takes time for the no flagella, the monotrichous, and the amphitrichous to become peritrichous. And it takes a little time for the lophotrichous cell to become peritrichous. Of course, you could have a species which is only lophotrichous, in which case you will see amphitrichous, monotrichous, and then no flagellas, but you would not see peritrichous. Any question about any of that? All right, if the cell is motile, meaning the cell can rotate or swim or run or tumble, then it has a flagella. Assuming it's a bacteria cell, it has to have a flagella because it's the only structure prokaryotes have to move. Taxis is the movement toward or away from a stimuli. In cells, we have 2 types of taxis. Chemotaxis, where the cells respond to a chemical, and then phototaxis, where the cell responds to the presence of light. Photosynthetic cells are phototaxic, and they usually swim towards the light. This cell is not photosynthetic because it's swimming away from the light, but it is showing phototaxis. Any question about any of that? All right, a special structure found only in the spirochetes is called an axial filament, or an endoflagella. This is a flagella, but this flagella is inside the cell wall. It's actually part of the cell wall. It is anchored at one end of the cell, and then it wraps around the cell. It gives the cell its characteristic spiral corkscrew movement. And here, let's blow that up just a minute. Come back here. Blow that up. Blow that up even further. You can see the cell wall is shown in light pink. The axial or endoflagella are shown in the red. And the cell wall goes around the endoflagella, meaning the endoflagella are inside the cell wall. Any questions about any of that? And there you can see the cell. Put that there. The cell can change its direction it's swimming in just by rotating the cell in the opposite direction. It'll cause the cell to move one way or the other. And there, you saw how flexible the spirochete is. It actually changed from running north and south to moving east and west. Let's look at that again. There. And it's moving in different directions just by changing its corkscrew rotation. Any question about any of that? Another example of the spirochete movement. It's thought that the spirochetes can actually burrow through mucous membranes. So its movement can burrow through mucous membranes. Any questions about any of that? All right, fimbriae are shorter, straighter, and thinner than flagella. They're also smaller than the sex pilus. They're used for attachment to surfaces. They are not used for swimming through liquid media. But the cell here can attach to something by the fimbriae attaching to it. And then let's talk about the sex pili. It is a structure similar to the fimbriae. Let me blow that up. This cell actually has shorter fimbriae right here, many of them, and then a sex pilus. That sex pilus is coming out of this cell, and it will literally skewer this cell, and then it can act like a winch to bring the two cells together. So two cells do not need to come real close to each other because the sex pilus is actually hollow, and then the plasmid can leave this cell and then enter that cell. The plasmid actually codes for the production of the sex pilus. And the sex pilus is used to transfer DNA, a plasmid, from this cell to that cell. Any question about any of that? If not, I'm going to stop the slide here and we'll continue at 7 o'clock with the lab. Any questions? All right, I'll say goodbye and see